Morning, everyone. Thanks for coming. I have a stationary microphone, so I can't walk around the stage because I wanted to point out that I've seen a few people with like cannabis socks or t-shirts or jewelry, and I don't have any of that. But I just wanted to point out I might not have the nicest trousers, but they're made out of hemp, so that's my nod to the industry. And they're made in Patagonia, which by Patagonia, so that's what down the coastline here. So getting all the commercial tips here. So today, when I applied to this to speak here, I wanted to talk about product delivery, like what do you actually consume? Because in the cannabis industry, you test the products, but you don't really test what ends up with the customer. And I had the idea of doing skin permeability tests and tell you how much comes through the skin. And research always takes longer than it should. But the other aspect is inhalation, right? There's a combustion or vaporization of compounds, so you don't actually know what you consume if you just test the oil or the joint that you do. And this is far out point that you apply to speak, and you never know where you get with your research or what is the current topic. So it turns out this is really timely. So we are focusing today on the current research on vaping, and it's really the research only, so what is known, and I try to stay away from speculation. But to make the point that this is timely, these were just a few cutouts and screenshots that I caught to make this talk, and then a few statistics behind it. And it turns out I had to send these slides on Friday, so everything is outdated. So as I saw yesterday, we had 530 confirmed cases, and we had 8 deaths now through the vaping problem. There could also be the thought maybe we didn't know what it was and we hadn't seen it. We had seen it but not realized what it was. But from the reports that I read and the literature that came out recently, this is a new case, or these are new symptoms. So it's not something overlooked, but it's something relatively new. So everyone is trying to figure out what it could be. So as far as I'm aware, there's no single cause yet identified. There are a few ideas what it could be. So I put up a few molecules at the bottom. So vitamin E acetate is the one that is highly discussed now, particularly through the New York statements. I also put up MCT oil. It's another stretching agent in the cannabis industry. And then PG and VG, more from the nicotine side. Because anyone who done formulation in the cannabis industry will realize they don't actually mix with cannabis oil, and you will see a separation after a few days. So if you sell them quick, you're good, but they will separate. And then just for popular request, we have popcorn lung here as well. So diacetyl, the buttery flavor that is not yet seen in the vaping in the cannabis industry, but it's at least in the vape, or was in the vape industry around. And I point out all these compounds are GRAS, because I heard this a lot over the last years when they talked about vape oils, ingredients, and it's like, it's GRAS, it's safe, it's good. So I put up the statement by FDA, and I counted there's 4 times the word food and not a single time the word smoking or inhalation. So these compounds are done for eating, ingestion, not for smoking. Thanks to some strong strong forces on the regulatory bodies, there's very little regulation on smoking. So that's the best we can go with right on the GRAS side. So on the GRAS side, I looked up on the FDA side, we are currently at 859 compounds that are on the list. So GRAS generally recognized as safe. So what is on the list? Or what used to be on the list is trans fats. So the list by itself is not perfect. And then secondly, I came here on Sunday. I came from Vancouver where it's raining and it's cold, and I came here and it's sunny and beautiful, so I went surfing in Malibu. And if Baywatch taught us one thing, it's not good to inhale seawater, but it's a GRAS compound. So I think I can make the point that GRAS is not per se good. and definitely not a good idea for inhalation. And then not GRAS. I looked it up because so many people told me butane extraction is good, it's safe, and if you inhale some butane, it's GRAS. So I looked it up, it's not there. No butane, no pentane, no hydrocarbons, it's not mentioned. So that's just the side one on extraction. So, but focusing on what I could find in the literature about inhalation. And by the way, I will just rumble on because my timer never started, so we will just go until the afternoon on this. Still not started. Anyway, so I found one paper from a German group. They were concerned with how much THC do you inhale if you smoke a joint compared to a dab. And I think that's an interesting aspect to start with, because so I assumed this is CannMed, so my audience is mostly doctors, so we are thinking about how do we treat patients, how do we give anything to the patients, how do we even recommend what they should consume. So let's start with how much cannabis or THC would you even inhale if you recommend a different consumption method. And so here are these rigged setups, and they first showed that in smoking and dabbing, there's a complete decarboxylation. So even if you have any THC acid in the product, in your product you consume, you will inhale THC. There is no THC acid coming through. And then secondly, and that we have to point out, is you do not inhale the gas or the vapor at temperature of combustion or vaporization. You inhale something around 44 degrees on average, maybe up to 60 degrees. Right? No one can inhale. So for you Americans, that is 111 Fahrenheit. So you could not inhale anything at 150 degrees Celsius. That would just burn your lungs, literally. And side note, the boiling point of THC by the literature, if you do the conversion, is 425 degrees Celsius at 1 atmosphere. I've seen the number of 157 around a lot, which is correct if you do it at reduced pressure, at 5 millimeters of mercury, but at 1 atmosphere it would be 450, and you definitely do not inhale, therefore, any THC vapor. You would actually inhale small goblets of oil that are coming through. So that's why you can't use normal cigarette filters in cannabis smoking, because they would catch all the oil droplets and you wouldn't get anything. And the understanding that oil droplets are inhaled is important for a second talk that comes after the break by Kyle when he talks about heavy metals, because gaseous heavy metals is not what you're inhaling either. Okay, so next point. The other point was recovery, or how much THC you actually inhale. And here in the joint they got to 27% of the THC that was in the joint actually ended up in the simulated lung, and for dabbing you went at 75% recovery. So if you think about treating a patient and you're switching them from joints to dabs, you can't just take the math and it's like, oh, that much THC you used to consume, now you do a different method, we just use the same amount. So you have to adjust there. But this study had its limitations because it was, as you can see on the picture, a pretty rigged system of how you inhale. They actually sucked the joint through in one go. They didn't do puff-by-puff studies. So if we look at other research, puff-by-puff studies, this is a set of graphs or diagrams for nicotine inhalation. I hope you can read this all well. So what these graphs are showing is the nicotine amount that is in the vapor per puff in different methods. So top left is a vape cartridge. So the nicotine level stays consistent. Because you have an oil, you have a homogeneous mixture, you always get the same load every puff. But if you have these heat not burn systems, that's bottom left, you have a change in nicotine concentration with every puff, which is opposing to the reference cigarette, which is the bottom right, where the nicotine concentration increases. And the same principle somewhat applies to smoking a joint, so that you start off with a low concentration of your active ingredient and it slowly increases. So here's a study of smoking a spliff from Germany. So I had to learn what a spliff is. In Europe, they don't consume straight up cannabis in a joint. They use a spliff, so it's a certain amount of nicotine, of tobacco together with cannabis. The reasons for that are unknown to me. It might be that we have a less supply of cannabis in Europe, or that they like the nicotine aspect to it. But what you see is THC, CBN, so that's an old joint, and nicotine start off at low concentration at the first puff and then slowly increase. The reason for that, you could think about it, because the burning of the joint or spliff is at the front, and it goes through the whole system before you inhale. So if anything burns and then goes through the system as a droplet, it might just be caught. So while it evaporates, it never makes it to your mouth, it just gets caught, and therefore is a buildup of higher nicotine and THC concentration later on in the joint or spliff. And then when you get to that, then you can inhale more. So that's why it's increasing. But it's not only THC, nicotine, and so on, the compounds you measured that you actually inhale. There are a few more other compounds that happen and are produced when you vape or combust the material. So as a chemist, this slide is cool. Not many chemists in the audience, I guess, so just be scared, so let's move on. So here I have a picture of a different setup. Looks way more sophisticated than the first setup. So this is now from a third group here. This is actually from a group in Portland. So they are hooking. Did you see the terpene talk with GCMS headspace analysis yesterday? So that's pretty cool. That's very similar to this. So this is also a GCMS headspace, so to say, where they smoke a cannabis sample, inhale it into the GCMS, and then measure in the MS what comes out. And while I already early on mentioned there are limitations to all studies I present, in Germany they had a police officer standing there watching the spliff while it gets smoked so that it doesn't get lost, because that was a cannabis spliff they found of someone when they busted him. Here in the US, you can't get any cannabis products into a university and do some research on it. So what they did was they bought a milligram of THC in reference standard, evaporated the solvent, put that milligram onto a test tube or so, and then smoked that milligram. And then looked at what compounds actually came out. And it was not only THC. So this is pure THC being vaporized and inhaled into the GCMS. And so we see isoprene units, we see formaldehyde, we see other ketones, we see aromatic systems on the other scale. So this already looks complex, and you can point out a few potential harmful substances coming out. But if you have a real cannabis sample, and we're jumping back to the German group here, there are way more compounds in there if you have cannabis, because, and I think I pointed that out on, no, I didn't. So way more compounds coming out. And there you can now also see the parent THC compound and CBN compound. So let's point out a few compounds. Isoprene unit, bottom left. It's the mother of all terpenes. So they basically just, while isoprene build terpenes, and then eventually cannabinoids, when you combust them or heat them, they can go back. And then some carcinogenic aromatic compounds. What we have to point out is that cannabinoids, terpenes has similar biosynthetic heritage. And secondly, with the more terpenes there are, the more volatile organic compounds you have, so the more stuff can happen. And can be formed. So if you then dig it a little deeper, and you now look at a few harmful compounds and wonder how are they produced at different consumption methods. So we have a dabbing, so of pure THC, THC terpene mixtures, that is this SND dab, and then vaporization at different voltages. So voltages here are a stand-in basically for the temperature of the heating element. The more electricity you run through it, the hotter it gets. It's not the perfect system because all batteries or heating elements are slightly different, but it gives you a good indication. And those numbers basically indicate the more voltage you send through the vaporization mechanism, the more of those decomposition products you get. And if you do dabs, you really have a lot of those. Your dabs are in temperatures considerably higher than any of your vaporization methods. And then on the risk factors, you cannot try to do like a theoretical calculation of how risky it is to do a consumption method. Like how bad could it be? This is not an absolute measure, and it is based on data that is in the best case indicative of real consumption, because these are based on consumption methods on a machine, right? The GCMS we saw, and as National Institute of Science pointed out, there is currently no indication that cannabis smoking or vaping actually leads to lung cancer. So they are good for comparison, not for absolute. So here are the numbers. The HI is for non-cancer risk and the ELCR is for cancer risk. And when you look at the numbers, you see that smoking flower is potentially the most harmful because you have a combustion of plant material with a lot of compounds that you could potentially inhale. And then we go down the list, dabbing, and then low voltage vaping would be the potentially less riskful. Doesn't mean that any of them are leading to cancer or that none of them lead to cancer. It's just a comparison between the methods. And then another slide I could add to this was one of the reports I saw that talked about synthetic cannabinoids as an agent in CBD vape cartridges. CBD is currently the craze. The supply of CBD might not be as high as it has to cover all the demand. So people were using other cannabinoids to stretch, or synthetic cannabinoids. And I want to point out, as a chemist, there is a difference between synthesized THC and a synthetic cannabinoid. They're not the same thing. So synthetic cannabinoids here shown on the left by basically one of the godfathers of the thing, John Hoffman from Clemson University. These were molecules made to target the endocannabinoid system. So they designed molecules that would hook onto a CB1 receptor in a Petri dish and shut it off to give a perfect response, right? These things bind and don't leave. They were never designed to actually be consumed by humans because they are full agonists or total or super agonists, while THC or CBD shown here are partial agonists, so they actually leave your brain at some point again. So I just wanted to point this out. It is often miscommunicated in the industry or misunderstood in the industry and miscommunicated in the press that synthetic cannabinoids and cannabis are like two different worlds. I think spices, they are referred to as spices, and bath salts are synthetic opioids. Both are available at your local gas station. So I just want to point out again some of the limitations. So this research is all very early on. The availability of real samples to do tests are limited. I know Kyle will talk about some research he actually did on real samples. If you do a bit of a non-official university study, that is possible, but if you do go through the normal routes, it gets a bit more complicated. And then another problem seems like a missing collaboration of different actors. So Health Canada is interested in this, universities are interested in this, producers are interested, but somehow they don't really work together. So everyone starts from scratch and builds it up themselves. It would really help if people would work together and allocate resources and potential better. So I have a few minutes, so let me plug my company. So our idea is fundamental collaboration. So we are a research institute or research facility that is full aware that we don't understand everything. So I'm a chemist, so I showed you the molecules. I have no idea what they do in a body. But for that, I have friends that know that aspect, and I have friends that know how to formulate vape products or other products. So we try to collaborate as much as possible. So as I said, our focus is on cannabis chemistry. And to point out this collaborative aspect and the understanding that we don't know everything, my team started to compile a list of compounds that are known in the cannabis plant. And we slowly start adding decomposition products like CBN that are not naturally occurring but are found in cannabis production, so that we can then start tracking them and then start assigning values of interest to them. So in our case, we start tracking molecular weight, melting point, solubility in different different solvents, and so on and so forth. So we had 24 compound categories, over 700 compounds. I know we can get to 1,000. Let's just give it some time. While we have 75 cannabinoids on our list, I know Professor Mary, who I very much admire, has more already in his list, but we're slowly trying to catch up. We had 400 terpenes. Yesterday we had like 200 terpenes, so I have you beat on that. And then the other aspect is this database is available to collaborate. So if you have some data sets that you want to add to it, and if you want to get some other data sets that we have, get out of it, we are very happy to collaborate. Please reach out to us. I want to have this list filled as quickly as possible so that we can have our metabolomics database of cannabis-related compounds. I know they're existing for other research fields. And with that, I have to thank the authors and researchers on the work I presented and the collaborators that helped me make sure that these slides are all accurate. And I'll put up one of the email addresses of the PhD student that did a lot of the inhalation studies, because you will have questions that I cannot answer, so please put his email address down and reach out to him. Also, if you want the slides, they are up on my website. I upload them all on SlideShare, so you don't have to take as many pictures of those slides. Then talking about pictures, I don't know why they put this picture of me in there. It's not the best one. At least it points out that I like to ride bikes. And here, these were the people that took the pictures of me and that helped me through this. So this is my current, or it's actually my former co-op crop. I'm faculty at UBC, so I get access to quite a lot of motivated students that come through my lab on a 4-month basis. They do one of their semesters, they have to work for a real company. So I pretend to be a real company. They work for me. So some of them have now changed, and I have now a new set of people. I added a statistician to the team so that we can get our numbers run better. And with that, thank you very much for listening. And if there are any questions, I think we have time during the now break before we all move over and listen to Carl's talk about heavy metals and vape cartridges. Thank you. Any questions? Yes. Yeah, so the question was, are there more compounds you inhale if you smoke a joint versus if you vape an oil? I would very much say so, yes. First, there are more compounds initially that you could even inhale, and then using combustion always produces way more compounds down the line than just vaporizing them. So one of the studies I showed you was actually on German material combusted. So there was a plethora, and there were way more compounds they found there versus vaporizing. And I hope next year I can add my own data to this. Oh, beneficial compounds. Ooh, beneficial. So I don't know generally if they're good or bad, the compounds, so I can't really answer this. That actually comes back more to my expertise of extraction. I would say that most extraction artists don't really know how to extract, so they might leave behind a lot of compounds that should be in the oil. So generally, I would say the oils you inhale that are available on the market are of lesser quality than they should be if you would extract correctly. Also, we all are really happy about the synergistic effect, and then we use distillate. So, like, pick one. Okay, what's a correct extraction? So we're veering off my just presented topic, and we're getting to the stuff I really like. So my argument is you have compounds in the starting material, and you want to get all of them out, and none of the others you didn't want. So if you have to clean up your extract with winterization and distillation, you didn't extract correctly. And if you're producing, if you have 10% yield on a 20% THC flower, you also didn't extract correctly because you left a lot behind or lost it during the process. So that's a short answer. Yeah, I lost my slide, so yeah. So nicotine can't break down into benzene because there is no benzene moiety in the molecule. That not said, if there are any other compounds that could produce benzene, that is beyond the reading I've done. I don't know the exact formulations of nicotine-based products. Interesting aspect, but I don't have any answer to that right now. There were another question?
Saying that vaporizing the oil, you're actually inhaling droplets of oil, not cannabis vapor, not THC vapor.
Yes, you have to, because
Probably most of the compounds are not volatilizing, you're actually inhaling droplets. My question is, what can you say about bioavailability of apples? What actually crossing the blood-brain barrier compared to smoking a joint, compared to
I don't know. I have to admit, I don't know. The reading I did for this presentation and the research was based on like which compounds come through. How they get then that goes into a direction that I'm not really qualified to talk about. There is the understanding that the smaller droplets are faster absorbed, I guess. I'm hoping to a nodding head. I think there was a nodding head. So. I'm trying to hook up a particle size analyzer to my vape machine right now, so that I can actually start doing that work, but we're not there yet. So for the study that was the GCMS study that I showed you, yes, that would be vaporization because they're putting pure THC in a tube, so they're not putting that into a vape cartridge. They only have 1 milligram of it. As I said, there are limitations to all these studies that are currently out there because no one yet has gone to a dispensary, bought a vape cartridge, hooked it up to their personal battery, and then put it into a machine and checked that. Yeah, so I don't know the answer. I'm aware of the question, so that's why I said I want to hook up a particle size analyzer to the study. So ask me next year, and I give you some data back. Oh. Did everyone hear this? Monica has a loud voice. She was one of the people that I mentioned in the slides because she knows a lot more about vaping specifically than me. I kept ignoring one follow-up question here on the front. Do you still want to ask this? Yes, so I had this slide earlier, but that was on nicotine side, so heat not burn is exactly that. You have flour in a compartment. You can either have a prong that sticks into it, that's the IQOS, or you have like a small jar of ceramic, and you put your flour in there. So yes, they behave very different to normal nicotine cigarettes, as I showed for the prong size. Not seen any data on cannabis itself for that. But it's definitely an interesting thing to build. And it has already been built, if I look in the right direction. So I'm sure PAX would be happy to discuss that on their stand at the expo. One quick question on the back.
You showed us some degradation, molecularly glycol, some of the other disorderly excipients, so that's interesting. But what's your next step? To link what's going on with these vaping deaths and diseases, and what are you going to do with it?
I live in Vancouver, Canada. I'm just happy that I'm not in the US. So no, I like a private research lab will never have the capability of figuring out what the reason for an epidemic is. We don't have access to enough samples, so we can't do this. What will be our focus is helping producers in the future to design their vape oils and hardware so that the inhalation leads to, or that at inhalation you have a desirable molecular profile, whatever that will be. But figuring out what an epidemic is caused by it can't be done by a private research lab. If they want to send me samples and I look at it, maybe, but I leave that to the professionals. I don't have a time. We all have to make it to the heavy metal talk. So thank you very much again. And just find me in the corridor afterwards if you have further questions.